This purple thing here is the helicase, which is an enzyme that's used to separate the two strands of DNA. Here, the single-strand binding proteins come to hold the two separated strands of DNA apart. Here we see the DNA being catalyzed by the polymerase. Now what's happening is the polymerase is adding new nucleotides and attaching them to the already existing template strand, creating phosphate as a waste product. Here the primase was laying down RNA primer. It did this because the DNA polymerase 3 can only replicate DNA strands from a 5' to 3' direction on the leading strand.
The leading strand is the strand complementary to the DNA template strand. The lagging strand, which is named so because it actually lags behind the leading strand during synthesis, is elongated in segments, not continuously. Here, a primase adds another RNA primer to elongate the lagging strand, as again, it is elongated in segments. Then, a DNA polymerase 3 synthesizes the RNA segment to the strand. A DNA polymerase 1 replaces the RNA with the DNA, adding to the lagging strand. Here, a DNA ligase joins the segments to the sugar phosphate backbone into a continuous DNA strand. Here, DNA polymerase 3 is synthesizing both the leading and lagging strands, the leading being synthesized continuously, the lagging being synthesized in segments. In the picture shown earlier, the DNA polymerase 3 proteins may seem to move along the leading and lagging strands separately, moving along the DNA like a railway. But actually, all of the processes are connected by protein reactions into the complex shown, with the DNA running through the complex.